Assalamu alaikum. Today we want to talk about part 2 in this chapter 21, Reactions of Monosaccharides. Now, reaction with oxidizing agent, reducing sugars. Aldehydes can be oxidized to carboxylic acid, RCHO, all the way to carboxylic acid. As the open chain aldehyde is oxidized, its equilibrium with the cyclic form is displaced so that the open chain form continues to be produced. Now, carbohydrates that react with mild oxidizing agent are classified as reducing sugar. You see alpha-D glucose, Howard form, Fischer, with oxidizing agent. The aldehyde group is oxidized all the way to carboxyl group. Now, in basic solution, however, ketones can undergo rearrangement and will also act as reducing sugars. Ketoenol tautomerism is an equilibrium that results from a shift in the position of a hydrogen atom and a double bond. It is possible whenever there is a hydrogen atom on a carbon adjacent to the carbonyl carbon. So here ketose in basic medium, it is converted to indiol. Still, it tautomerizes to the keto form, and still oxidizing agent, it goes all the way to aldonic and anion. So again, ketones can undergo rearrangement and will also act as reducing sugars. This is how ketones can undergo rearrangement and act as reducing sugars. Reaction with alcohols, glycosides and disaccharide formation. Hemiacetals react with alcohols with the loss of water to yield acetals, compounds with two OR groups bonded to the same carbon. Here, this is a hemiacetal plus alcohol, acid catalyst to form acetal زائد water. Because glucose and other monosaccharides are cyclic hemiacetals, they also react with alcohols to form acetals, which are called glycosides. So here again alpha-D glucose plus methanol, the presence of acid catalyst, it forms this methyl alpha-D glucoside and acetal. And this is known as a glycosidic bond. In larger molecules, monosaccharides are connected to each other by glycosidic bonds. Here you see formation of a glycosidic bond between two monosaccharides. Here we have monosaccharides by loss of water molecule. We form an alpha glycosidic linkage here. The reverse of this reaction is hydrolysis. Hydrolysis of disaccharides with water yields the two monosaccharides. Formation of phosphate esters of alcohols. Phosphate esters of alcohols contain a phosphate group bonded to the oxygen atom of an OH group. The OH groups of sugars can add phosphate groups to form phosphate esters in the same manner. Phosphate esters of monosaccharides appear as reactants and products throughout the metabolism of carbohydrates. Glucose is phosphorylated by ATP in the presence of enzyme called hexokinase to form glucose 6-phosphate. And this is the first step in glucose metabolism, as we will see in the coming chapters. Now, disaccharide structures. The two monosaccharides in a disaccharide are connected by a glycosidic bond. Now, the bond may be alpha or beta, as in cyclic monosaccharides. Then here we have, this is carbon 1, this is carbon 4, and this is called an alpha 1,4 acetal link. This is carbon 1, this is carbon 4, but it is up. It is a beta 1,4 acetal link. So the structures include glycosidic bonds that create a 1,4 link between carbon 1 of one monosaccharide and carbon 4 of the second monosaccharides. Maltose can be prepared by enzyme catalyzed degradation of starch. Two alpha-D glucose molecules are joined in maltose by an alpha 1,4 link. It is both an acetal at carbon 1 in the glucose on the left, and hemiacetal on carbon 1 in the glucose on the right, making maltose a reducing sugar. So this is a glycosidic bond at carbon 1, this is the alpha 1,4 linkage, and this is the hemiacetal at carbon 1. So maltose, which consists of two glucoses joined together by alpha 1,4 linkage, is a reducing sugar. Lactose or milk sugar is the major carbohydrate in mammalian milk. Human milk, for example, is about 7% lactose. Structurally, lactose is composed of D-galactose and D-glucose. The two monosaccharides are connected by a beta 1,4 link. Like maltose, lactose is also a reducing sugar because the glucose ring on the right of the following structure is hemiacetal in carbon 1. So there is a free OH group. That's why lactose is also a reducing sugar. Sucrose, on the other hand, hydrolysis of sucrose yields one molecule of D-glucose and one molecule of D-fructose. Now the 50-50 mixture of glucose and fructose that results in vert sugar SAMU is sweeter than sucrose. Now sucrose itself has no hemiacetal group because a 1,2 link joins both anomeric carbon atoms, carbon 1 here on glucose and carbon 2 on fructose. These are the two anomeric carbons. This is an anomeric link. So the absence of a hemiacetal group means that sucrose is not a reducing sugar, remember. So we can say all monosaccharides and disaccharides except sucrose are reducing sugars. Notice here the type of linkage here. It's an alpha 1 beta 2 linkage. Alpha 1 beta 2 linkage. Variations on carbohydrates often incorporate modified glucose molecules. Here we have beta D glucouronate. Here we have beta D glucosamine. Here we have N-acetyl beta D glucosamine. Chitin. The shells of lobsters, beetles, and spiders are made of chitin, the second most abundant polysaccharide in the natural world. Cellulose is the most abundant. Chitin is a hard structural polymer. It is composed of N-acetyl D-glucosamine rather than glucose, but is otherwise identical to cellulose. Both chitin and cellulose contain beta 1,4 linkage, but the difference is in the monomer. The monomer in cellulose is glucose, and in chitin it's N-acetyl D-glucosamine, the one I showed you before. This is the monomer in chitin. Connective tissue and polysaccharides. Connective tissues are composed of protein fibers in a matrix that contains unbranched mucopolysaccharides. The gel-like mixtures of these polysaccharides with water serve as lubricants and shock absorbers. Hyaluronate molecules contain up to 25,000 disaccharide units. It forms the synovial fluid that lubricates joints and is present within the eye. However, chondroitin 6-sulfate, also the 4-sulfate, is present in tendons and cartilage, where it is linked to proteins. Then here you see the structure of the two glycosaminoglycans, hyaluronate repeating unit and chondroitin 6-sulfate repeating unit. They are heteropolysaccharides because this repeating unit contains two different monosaccharides. That's why they are, whereas chitin and cellulose are homopolysaccharides as well as glycogen. Heparin is valuable medically as an anticoagulant, an agent that prevents or retards the clotting of blood. Heparin is composed of a variety of different monosaccharides, many of them containing sulfate groups, as you can see here. So this is an example of repeating unit in heparin. Heparin is also of this family, glycosaminoglycan, and this is the repeating unit in heparin. Glycoproteins. Now, proteins that contain short carbohydrate chains, oligosaccharide chains, are glycoproteins. You see here, the carbohydrate is connected. This is inside of cell. This is the lipid bilayer. This is your protein. And attached to the protein is an oligosaccharide. This is outside of the cell. So this carbohydrate that is connected to a protein by a glycosidic bond between an anomeric carbon and a side chain of the protein. The oligosaccharide chains function as receptors or, in one case, antifreeze. Some important polysaccharides. Polysaccharides are polymers of tens, hundreds, or even many thousands of monosaccharides linked together through glycosidic bonds. Cellulose repeating unit, 2 glucoses joined by beta-1,4 link. Starch and glycogen repeating unit, 2 glucoses are joined together by alpha-1,4 linkage. But back to cellulose provides structure in plants. Each molecule consists of several thousand D-glucose units joined in a long straight chain by beta-1,4 links. Because of the tetrahedral bonding at each carbon atom, the carbohydrate rings bend up and at one end and down at the other in what is known as the chair conformation. This is called chair conformation. Then cellulose is a homopolysaccharide, consists of alpha D-glucose units joined together by beta-1,4 linkage, and it gives structure in plants. It provides structure in plants. Now, the hydrogen bonds within chains and between chains, the one you see, within chains and between chains, contribute to the rigidity and toughness of cellulose fibers. Now, grazing animals, termites, and moths are able to digest cellulose because microorganisms colonizing their digestive tracts produce enzymes that hydrolyze beta-glycosidic bonds. Humans neither produce such enzymes nor harbor such organisms, and therefore cannot hydrolyze cellulose. Because we do not have enzyme called cellulases, and cellulase is the enzyme that digests cellulose, but termites do. Starch. In starch, individual glucose units are joined together by alpha-1,4 linkage rather than by kada. Starch is fully digestible and is an essential part of the human diet. It is present only in plant material. Our major sources are beans, wheat, and rice, and potatoes. Then rice and potatoes are major sources of starch. Amylose accounts for about 20% of starch. It is somewhat soluble in hot water and consists of several hundreds to 1,000 D-glucose units linked in long chains. Amylose tends to coil into helices. So, amylose, linear, all the monosaccharide units are connected by alpha-1,4 linkage, and amylose accounts for about 20% of starch. On the other hand, amylopectin accounts for about 80% of starch. It has up to 100,000 glucose units per molecule, and alpha-1,6 branches approximately every 25 units along its chain. It is not soluble. Branch point in amylopectin, also in glycogen. You see here, this is the alpha-1,6. This occurs at branch point. The other linkages are alpha-1,4. Glycogen stores energy in animals in the liver and muscles. Structurally, glycogen is similar to amylopectin in being a long polymer of D-glucose with the same type of branch points in its chain. Glycogen has many more branches and contains up to 1 million glucose units per molecule. In glycogen, a branch occurs at every 10 glucose residue, whereas in amylopectin, a branch point occur at every 25 glucose unit or residue. That's why glycogen is much more highly branched than amylopectin. Amylopectin in plants, glycogen in animals. Cell walls, rigid defense system. Bacteria and higher plants surround the plasma membrane with a rigid cell wall. The rigidity of the wall prevents the cell from bursting due to osmotic pressure, gives shape to the cell, protects it from pathogens. Plant cell walls are composed of fibrils of cellulose and polymer matrix of pectins, lignin, and hemicellulose. Bacterial cell walls provide a rigid platform for the attachment of flagella and pili. They do not contain cellulose. A majority of bacterial cell walls are composed of a polymer called peptidoglycan. Animals have developed natural defenses that can control many bacteria. For example, lysozyme, an enzyme found naturally in tears, saliva, and egg white, hydrolyzes the cell wall of pathogenic bacteria. Then lysozyme is the enzyme that is responsible for hydrolyzing the cell wall of bacteria. Penicillin contains a beta-lactam ring that acts as a suicide inhibitor of the enzymes that synthesize the peptidoglycan cross-linking peptide chain. Unfortunately, many bacteria have developed enzymes that destroy the ring, granting resistance to penicillin. And from here till the end, this is called chapter summary. You can read it by yourself. What are the different kinds of carbohydrates? Why are monosaccharides chiral, and how does this influence the numbers and types of their isomers? What are the structures of monosaccharides and how are they represented in written formulas? How do monosaccharides react with oxidizing agents and alcohols? What are the structures of some important disaccharides? What are the functions of some important carbohydrates that contain modified monosaccharide structures? All these we talked about in the text. This is a summary. What are the structures and functions of cellulose, starch, and glycogen? And this is end of part 2.